Okay, students, Assalamualaikum warahmatullahi wabarakatuh and salam sejahtera. For today's lesson, we are going to learn the last topic for Form 5, okay, Chapter 13, which is Genetic Technology. So, when we talk about genetic technology, you have to understand that, okay, the insertion of DNA from other organism into another organism, okay, which are related to GMO, GMF, and then we have to relate with gene therapy, genome, okay? So you will learn after this more detail. Okay, first of all, look at the definition for genetic engineering. Apa maksud kejuruteraan genetik? So genetic engineering is a gene manipulation, okay? Alteration of gene. Okay, this technique very important to modify genetic material or genetic constituent of an organism so that new combination of genes can be produced. Okay, so organisms such as animal, plants, or microorganism that is produced by recombinant DNA technology is known as genetically modified organism, okay, which is GMO. So genetic engineering involves the transfer of a DNA segment or genes from one organism to another by DNA recombinant technology. So this technology will combine, okay, DNA from one organism to another organism. That's why producing recombinant DNA. Okay, so by using the techniques and procedures in the recombinant technology, biologists can recombine DNA or genome, a complete set of DNA for an organism. Okay, so this one example, organic tomato and GMO tomato. Okay, so what is a genome? I mentioned before about genome. So genome is one whole set of your genes plus all the DNA between your genes. Okay, example given here, the human cell, okay, your cell. So most cells in the human body have a complete set of genes. Okay, you can see from bio exploration in your textbook, a complete set of DNA for an organism that includes all the genes of that organism. So the genome contains all information needed to build and carry out the life processes of an organism. Okay, jadi genome ni merangkumi keseluruhan jin yang ada dalam tubuh badan awak. Termasuklah semua DNA dalam sel. Okay? Okay, students. Look at the genetically modified organism, GMO. So the GMOs are organisms that contain recombinant DNA or rDNA. Okay? So recombinant DNA technology will enable production of new gene combination. So, melalui teknologi DNA recombinant ni, saintis boleh combine DNA daripada satu organism dengan organisma yang lain dan menghasilkan recombinant DNA, new gene combination. So, organism that contains rDNA is known as transgenic organism. Example given here, okay? So this goat, genetically modified goat, possess the human gene. Okay, so the human gene which codes for blood clotting factor. So this blood clotting factor or blood coagulation factor found in the goat's milk. Okay, dalam susu kambing ini ada agen pembekuan darah. So agen ni, okay, the blood clotting factor can be purified. Boleh diambil, boleh diekstrak. Okay, kenapa? Untuk mengubati pesakit hemofilia. Treat hemophilic patient. Okay, Next, this genetically modified cow. Okay, lembu pula kat sini. The milk does not contain beta-lactoglobulin. Okay, this type of protein normally will cause allergy in some children. Okay, kanak-kanak ada yang memang tak boleh terima susu lembu just because of the presence of beta-lactoglobulin. So, scientists alter the content of the milk. Okay, so that beta-lactoglobulin is not present in the cow's milk. Okay. Next, look at the genetically modified food. Okay, tadi kita tengok organism. Actually, this one subset jugalah. Tapi food ni lebih kepada apa yang kita makan dan daripada sayur-sayuran, buah-buahan. Okay. So, recombinant DNA technology successfully produce varieties of crops. Okay, tanaman terutama. Agensi yang banyak buat research berkaitan GMF ni seperti MARDI, RISDA, MADA. Okay. So examples, paddy, okay, pokok padi, oil palm, kelapa sawit, pineapple, corn, soya bean, and livestock. Okay, ternakan seperti ikan salmon, cattle, and goat. Okay. So GMF will possess DNA from other species of plant or animal. So, binatang ataupun buah-buahan, sayur-sayuran yang terima jin atau DNA daripada organisma lain, dia akan tunjukkan DNA yang baru. Okay, kita tengok nanti contoh-contoh. But consumption of GMF still unknown. Okay, long-term effect still unknown. These are the characteristics of GMF. Okay, the first one, resistant to herbicide. Okay, rintang terhadap herbicide. Okay, next, resistant to disease. Okay, application in medicine. Okay, tolerance to heavy metals and resistance to pests. Okay. These are some examples of top 10 GMF, the corn, soy, cotton, papaya, rice, okay, canola, potatoes, tomatoes, dairy products, and peas. So please, kalau awak beli apa-apa juru makanan, awak boleh check dekat ingredients. Dia akan mention sama ada itu adalah GMF atau GMO. Okay. Okay. Now look at the advantages and disadvantages of GMF. Okay, the presence of GMF actually can overcome worldwide food shortage. Sebab kenapa kat sini? Melalui GMF boleh hasilkan transgenic crops dan juga livestock yang lebih kualiti. Dan masanya biasanya lebih singkat. Okay, reduce cost of food production. Okay, increase nutritional value of crops, reduce problems of crops related to pests, okay, reduce the usage of pesticides, increase in production will reduce price of food. Thus, this will increase the food availability. Okay, for disadvantages, for sure this one, endangered natural species. Okay, the natural one will become extinct. Okay, so there is a slight possibility that the foreign gene in GMF may be transferred to humans. So when we eat the GMF, there are possibilities that the genes will be transferred to our body cell. For example, antibiotic resistance gene. So kita makan makanan yang memang ada antibiotik dimasukkan ke dalam GMF tersebut. Jadi ada kebarangkalian untuk masuk dalam badan manusia. Okay, and adverse effects on human health and genetic material. So long-term effect still unknown, tapi memang memberi kesan kepada kesihatan termasuklah kandungan genetik yang ada dalam badan manusia. Okay. So, please check the ingredients. Apa saja yang awak beli? Know your labels. Okay, G, or GMF. Okay, so boleh nampak kat sini 5-digit code starting with 8. Okay, for organic 5-digit code starting with 9. Okay, and conventionally grown 4-digit code starting with 3 or 4. Okay, that's why the code kalau beli buah-buahan tempatan yang memang dijual dekat pasar raya ataupun yang diimport. Kalau jual dekat tepi jalan semua tu biasa tak ada kod lah. Jarang ada kod. Okay, buah-buah yang ada kod ni biasanya buah-buah yang memang ditanam secara meluas. Maksudnya dekat kebun dia ada, dia memang well organized lah. Okay. Okay, example given, this one. Okay, the corn produced by recombinant DNA technology. How? Okay, so Bacillus thuringiensis, okay, gene from this bacteria actually have been inserted to the corn. So that this will increase the resistance against insect pests. Okay, untuk lawan serangga. How actually? Bila dia makan, dia akan mati. Okay, kita tengok kat sini. Crop is infected by European corn borer. Borer maksudnya dia boleh menyebabkan lubang-lubang pada jagung tersebut. Okay, corn tersebut. Sebab dia makan kan. Tapi, pest tu akan mati when feeding on any plant parts. Just because of the gene from Bacillus, okay, inserted to the corn plant. Okay. So, bagi kesan, dia akan mati. Okay. Next, super salmon, okay, genetically modified by the age of 8 months. How? Okay, this type of GMF modified and approved by FDA. Maksudnya worldwide memang approved, okay, as a safe food for consumption in US and Canada. So modification involve insertion of growth hormone. Jadi masa usia ikan salmon ni 8 bulan, okay, growth hormone, hormon pembesaran dimasukkan, gene yang kod kepada hormon pembesaran lah. Okay, so the gene inserted codes for growth hormone, okay, into the genome of Atlantic salmon. So apa kesannya? Super salmon created which grows at a faster rate. So at the same time, the super salmon also can be produced throughout the year. Sepanjang tahun boleh dihasilkan. Okay. Next, potato. Okay, effect of frost on potatoes. Nampak mudah sangat oxidation berlaku dan hitam. So potato is an important crop to people who live in cold climates. Okay, but it is very sensitive to low temperature and frost. Jadi sejuk sangat ataupun beku bagi efek kepada potato di sini. So gene from Arabidopsis plant, okay, enable the plant to tolerate freezing condition. Okay, so gene from this type of plant inserted to the potato genome to create a genetically modified potato, so that this potato can grow well in cold climates. Jadi, ubi kentang ni mudahlah nak hidup kawasan yang bersalji dan sentiasa sejuk. Dia takkan jadi warna hitam lah. Okay. Next, look at the production of insulin. Okay, so you have learned in Form 4, insulin is an important hormone that will control the blood glucose level. Okay, so insulin will be extracted actually from pancreas of cattle or pigs to treat diabetes mellitus patient. sebelum ni ada syubhah, kalau dalam Islam syubhah, ataupun ada keraguan sebab insulin tu diambil daripada khinzir. Okay, so now insulin actually can be commercially produced by genetic engineering for patient with diabetes mellitus. How? Okay, kita tengok nanti prosedur yang terlibat. Okay, kita tengok kat sini. So, a spliced human DNA with insulin extracted or cut using restriction enzyme. So, the gene that codes for production of insulin isolated from human. Okay, and you see here the plasmid actually a small circular DNA from bacteria or virus. In this case, bacteria. Okay, circular DNA found in bacteria used as cloning factor or cloning vector. Okay, agent ataupun dia yang akan bawa jin manusia ni after recombinant DNA, okay, carried out. Okay, the technology carried out, you can see from here, a cut plasmid by using DNA ligase enzyme. Ligase means they akan attach the gene from human with the plasmid from bacteria. Okay, enzyme ni yang akan bertindak untuk lekatkan plasmid, the circular DNA from bacteria, with the insulin gene from human. So, human insulin gene is inserted into a plasmid. So, now a recombinant plasmid formed. After that, the recombinant plasmid introduced into a bacteria. Okay, so awak kena faham, bakteria dia replicate sangat cepat. By inserting the recombinant plasmid, okay, so more insulin can be produced. Okay, bacterial clone multiplies and produces insulin, so more insulin can be extracted from transgenic bacteria and purified. So, dia akan extract keluarlah insulin yang diperlukan. Bakteria kita tak guna. Okay, so beside insulin, other success of genetic engineering include hepatitis B vaccine, blood clotting factor, and growth hormone. Okay. Okay, now look at the concept of biotechnology. So, biotechnology is a field which utilize or use technology or method to manipulate organism for the production of biological products. Okay, so biotechnology, the aim or the purpose is to improve the quality of livestock and crops as well as to develop the application of microorganism for specific purposes. Okay, kita akan tengok contoh nanti. Since biotechnology involve various discipline of science, so each discipline that involve the use of living organism for a specific purpose given a specific color code. Okay, this can be shown from this diagram. Okay, kita tengok nanti. Although 10 biotechnology activities are identified, okay, the main ones are the green, okay, the white, yellow, and blue biotechnology activities. Okay, so tengok kat sini, environmental biotechnology, okay, the white color, industrial processes and involve microorganism, okay, the yellow color, the food and nutrition, bioinformatics and computer science, and the blue biotechnology activity, marine biotechnology. Okay, so kita lebih fokus kepada kat sini lah. Okay, 1, 2, 3, 4, 5 cabang yang memang banyak ditekankan untuk biotechnology. Okay, other than that, you can see improving agricultural products, biotechnology of desert and dry regions, okay, bioterrorism and biological warfare, okay, law, ethics and philosophy, medicine and human health. So kita banyak fokus 5 ini. Compare dengan lagi 5 lah. Okay. So look at the applications of biotechnology in life. Okay, in terms of medical, gene therapy. Okay, what's the meaning of gene therapy? Okay, gene therapy actually how the scientists alter the defect genes. For example, sickle cell anemia. So, gene therapy important to treat or prevent genetic diseases. In gene therapy, a normal gene inserted into the patient to replace the abnormal gene. That's why correct the defective genes. Okay, so disease that can be cured by gene therapy are cystic fibrosis, muscular dystrophy, and a few types of cancer. Okay, ini hanya contoh saja. Okay, so you can see the procedures involve a cell with an abnormal gene. Okay, you can see this one, abnormal gene. Okay, so insertion of normal gene to replace the abnormal gene using a virus. So now the cell perform normal function. Okay, so the first successful gene therapy was performed in 1990. Okay, a patient with ADA-SCID, a genetic disease that interfered with her body immune system. So di negara Malaysia kita tak tekankan sangat. Kita punya teknologi agak ke belakang. So gene therapy ni salah satu yang advance, yang perlukan device yang lebih canggih lah. Okay. So you can see example given here, the procedures involved. Okay, the cells harvested or extracted from patient. Okay, so in lab, virus altered, so cannot reproduce. After that, gene will be inserted to the virus. So altered virus, okay, mixed with the patient cell, so the cell become genetically altered. So altered cell injected into the body of the patient. So altered cell will produce desired protein. Okay, general procedures involved. Okay, so now look at the DNA profiling or DNA fingerprinting. Cap jari DNA, bukan cap jari ini. Okay, so now more to the bands. Okay, that will be produced from the samples. For example, kalau berlaku criminal case, jadi tempat kejadian tu ahli forensik akan cari dulu sampel yang boleh diambil. Okay, contohnya semen, okay, air mani, the blood or hair. Itu contohlah yang biasa didapati dekat kawasan yang berlakunya kejadian, okay, jenayah. Okay, contohnya pembunuhan dan sebagainya. So DNA profiling is a technique used in forensic to identify individuals. So nak kesan suspek, pastinya kena cari juga bahan bukti. Ini yang paling tepat untuk tentukan siapa penjenayah, terutama apabila melibatkan pembunuhan. Okay, so based on their DNA. Why? Because individuals' DNA is unique except between identical twins. So identical twins memang sama, tak boleh nak beza kecuali cap jari. Okay, so a human DNA set is different from animals. Okay, so and individuals can be identified based on DNA obtained from blood, semen for male or skin. Okay, so the applications of DNA profiling or DNA fingerprinting identify criminal. Okay, settle paternity and maternity dispute. Contohnya kita tak tahu anak siapa. Nak tentukan DNA, so boleh tahu. Check paternity, siapa bapa anak ni, siapa ibu anak ini. Okay, identify genetic disease. Okay, and determine suitability of organ donor and recipient. Okay, compatibility of the organ. Walaupun berlakunya donation of organ, kena check dulu compatible ke tidak nak terima organ. Okay, bukan simply saya nak derma organ and then orang ni terima. Tak boleh, kena check dulu. Okay. Okay, you can see DNA profiling can be used as a strong evidence in a criminal case. Kenapa kat sini? Okay, if the biological sample, for example, semen, the blood, okay, obtained and processed correctly and consensusly, sangat jitu. Okay, tak berlakunya penipuan, sangat jujur. So forensic scientists test the sample and then analyze it in a transparent manner. Okay, so the test results are interpreted precisely and the jury and the judge will obtain an accurate report. Okay, example given here, the recipe sperm, okay, case rogol di sini, boleh dapat semen daripada tempat kejadian. So, kalau ada suspek, kita boleh extract DNA, okay, daripada suspek dan check dengan semen yang diperoleh daripada tempat kejadian. Okay. Okay, students. Now look at the steps used in DNA profiling. Okay. So now the blood, semen or skin sample obtained from crime scene. For example, crime scene. Okay. So DNA is extracted from the sample. After that, restriction enzyme will be used to cut DNA into DNA fragments. Okay, with different sizes. So DNA fragments now separated by using gel electrophoresis. Okay, so DNA fragments of different sizes transferred, okay, from the gel into nylon membrane, and radioactive probes are added to the nylon membrane. Okay, so X-ray film then placed on top of the nylon membrane and X-ray film then processed to show the position of DNA bands that form the DNA profile. Okay, so please memorize the steps and please understand the steps. Okay, so this one, southern blot. Okay, the mom, which is blue color, dad with yellow, okay, and 4 children. D1, the biological daughter, okay. Next, D2, the stepdaughter, the child of mom and her former husband, which is red, you can see, okay. S1, okay, the biological son, and S2, adopted son, not biologically related. So his parents are light and dark green. Okay, so actually gunakan southern blot ni, you boleh tentukan, boleh tahulah which one yang pair. Sebab tu kalau buat ujian DNA, paternity, maternity, boleh dapat 99%, kadang-kadang 95%. Okay, sama dengan ibu dan juga ayah. Okay, according to the bands produced. Okay, so based on DNA profile below, can you determine who is the biological father of the child? So just like calculation, awak tengok. Kena kira lah. Which one yang sama dengan father 1, father 2. Okay. Tengok child ni. Awak kena check. Father 1, father 2. Which one yang tally dengan yang warna merah ni. Kalau tak sama, bukan ayah dia lah. Okay. Mana yang lebih banyak sama dengan child ni. Okay. So are we related? Kita tengok kat sini, 8 is mom, 9 and 10 are identical twins, and 11 is dad. 12 and 13 are random dudes. So the arrows show bands that are in the twins but not the mom. So instead, you can find those bands in dad. Okay. So biasa keluar dalam exam, awak kena cari. Simple calculation saja, bilanglah mana yang sama kan. Okay. So whose DNA match the DNA left at the crime scene? Ini tak berapa nak clear. Actually, based on the bands produced, awak boleh detect suspek mana yang salah. Okay. So this is the victim. This is the crime scene. Okay. Nampak? Kita tengok crime scene. Sampel yang dapat daripada tempat kejadian. So yang paling teli adalah suspect 1. Okay. Okay students, now look at the production of insect-tolerant plants. Okay, kita dah belajar tadi tentang BT corn. Okay, corn inserted with the Bacillus thuringiensis gene so that the pest that eat on the corn will die. So now BT cotton. Okay, BT cotton. So farming cotton breed with high yield and resistant to pests such as BT cotton in cotton farms has reduced the use of pesticide. So bila kita alter kandungan genom pokok kapas ni, okay, pastinya ini akan mengurangkan penggunaan racun serangga perosak. Kenapa? Sebab kuranglah serangga yang akan serang pokok-pokok cotton ni. Okay? So the BT crop actually injected with genes from Bacillus thuringiensis. Sama macam tadi. Tadi corn lebih kepada makan, sekarang lebih kepada penggunaan kita untuk hasilkan baju dan pakaian kan. Okay? So BT, a type of soil bacteria that secretes toxin. Okay, toxin produced actually very harmful to the pest. Okay, so this effort have saved costs by reducing the use of pesticide. At the same time, this will increase the cotton yield. Hasil pun bertambah. Okay, so besides that, BT crops are corn and brinjal. Okay, saya dah terang tadi tentang corn, sama je konsep, and brinjal ni awak tahulah. Okay, terung dan sebagainya. Okay, so these are the steps involved. Gene from BT, okay, Bacillus thuringiensis, codes for formation of toxin produced by the Bacillus thuringiensis. Okay, itu maksudnya. So the genes that codes for formation of the toxin from Bacillus thuringiensis will be inserted into the plant's genome. Okay, so now the pest feeds on the plant which produces BT toxin. So the toxin affect the digestive system of the pest and the pest will die due to effect of the toxin. Okay. So look at the benefits of GMOs and risks of GMOs. Okay, apa faedah ataupun kelebihan daripada GMO dan kita tengok juga risiko GMO. Okay, so benefits of GMO including nutritional value of foods could be improved. Example, introducing proteins, vitamins or vaccines. Okay, next, crops can be produced that lack known allergens. Okay, kurangnya allergen contohnya production of wheat without gluten. Okay, gluten biasanya menyebabkan allergy. Okay, crops can grow in arid condition for better yield. Okay, by introducing drought-resistant gene, for example. Okay, pokok tu boleh tahan terhadap kemarau. So, GM crops can produce herbicides to kill pests naturally. So, tak perlu guna herbicide ya, chemical. Okay. Next, improve food supply, agriculture in poor countries. Okay, so GM crops can be engineered for improved yields. Next, GM crops may have longer shelf life. Okay, less spoil, susahlah nak rosak. Next, reduces economic cost and carbon footprint. So that less need for land clearing and pesticide usage. Okay. And the risk here, new traits could cause adverse health reaction. So new protein form due to new gene form. Okay, so this may cause allergic responses. Removal of traits also could have unknown effects. And crops may limit biodiversity of local environment. Okay, jadi awak pun tahu secara naturalnya dia akan limit biodiversity. Tiada kepelbagaian sebab kita dah alter. Okay. So increase competition with native species. And cross-pollination also could lead to super weeds. Kita boleh hasilkan satu tumbuhan, satu kumpulan tumbuhan yang baru. Super weed. Satu, just like mutant lah yang saya explain sebelum ni. Okay. So patents restrict farmers from accessing GM seeds because biotech companies hold monopolies over crop use. Okay. And the foods with GM components may not be labelled. Kadang-kadang tak ada yang di-label macam yang kita makan sekarang. Okay. And different governments may have conflicting regulatory standard concerning safe usage. Macam Malaysia, kita tak berapa tekankan sangat GMO ni. Ada negara, dia ada awareness berkaitan GMO dan juga GMF. Okay. Okay, now cleaning of oil spill. Okay. So, due to biotechnology, by using bacteria, bioremediation. This one is a method that will use bacteria to clean up environmental pollutants. For example, oil lah. Okay. Tumpahan minyak yang berlaku dekat lautan, boleh gunakan bakteria untuk clearkan minyak tersebut. Okay, very effective to treat oil spillage in the ocean. Okay, bayangkan takkan nak gunakan tenaga manusia nak clear minyak dalam lautan. Okay, so most molecules in crude oil and purified oil products can be degraded by bacteria. So bacteria used can break down the particles. Okay, so example here, Alcarnivorex borcumensis. Okay, a type of bacteria that depends on oil for its source of energy. Maksudnya dia akan consume oil tersebut. Okay, so what we use to treat oil spill? So next, look at the importance of biotechnology in life. Okay, biotechnology often associated with agriculture, industry, nutrition, medicine, and other fields. Rapid development in biology and technology replace the old methods, okay, with the current and sophisticated approaches in producing many types of products that benefit humans. Okay, besides contributing to increase the yields in food, agriculture, medicine, biotechnology also improve a country's economy, specifically in industrial sector. Awak pun tengok, bila ada kita apply biotechnology, boleh hasilkan nanotechnology particle, contoh penghasilan produk kosmetik. Ini memang sangat advance lah bagi Korea, Jepun dan sebagainya. Okay. Okay, lastly, contributions of biotechnology to life in agricultural fields. Okay, the contribution including produce the best livestock and crop breeds. Okay, jadi boleh hasilkan ternakan dan juga tanaman yang memang baiklah, terbaik. So the tissue culture method and cloning technique have expedited plant reproduction. So produce paddy varieties which give more yield and mature faster. Jadi pokok padi pun pelbagai jenis, banyak variety, banyak hasil, okay, dan lebih cepat matang. Animal cloning also enable mass reproduction and produces disease-resistant clones. Okay, so cloning ni boleh hasilkan pembiakan yang lebih cepat, okay, lebih banyak, and then boleh hasilkan clone yang tahan kepada penyakit. Okay, use of hormones will expedite animal growth and maturity. Jadi penggunaan contoh tadi growth hormone mempercepatkan pembesaran haiwan dan juga kematangan haiwan tersebut. Ini bagi agriculture. For medicine, okay, modify genetic contents of an individual. Kita dah terangkan tadi, saya dah terangkan tadi, okay. So next, reduce risk of mental disease, okay. Prevent specific disease, for example, cancer and inherited disease, for example, hemophilia, muscular dystrophy, and so on. And human dwarf genetic, okay, also can be reversed. Okay, kita boleh ubat, okay. So can have normal children by using gene therapy, and assist mothers with difficulty to conceive. So, seseorang wanita yang susah nak mengandung juga boleh diubati melalui bioteknologi. Okay, for forensic, okay, identify criminal based on tissue or body fluid, for example, blood, semen, skin, or hair follicle. Okay, and the last one, environment. Okay, microbes can expedite decay of waste products created by humans. Jadi penggunaan mikroorganism, contohnya bakteria, boleh cepatkan proses decomposition, proses perebutan. Okay, produk-produk contohnya plastik sekarang ni. Okay, yang dihasilkan oleh manusia sendiri. And microbes also can degrade feces completely. So, hasil tinja manusia, haiwan dengan menggunakan bakteria juga boleh decompose waste product tu dengan lebih cepat. And the last one, reduce pollution to protect humans and environment. So, contoh tadi, oil spill lah. Okay, so that's all for today. Thank you.